Hi everyone, I'm Dr. Preeti Sharma and I'm an educator on the platform of Unacademy. Well, I hope all of you have been following this series of microbiology starting from day one. So we've had a total of 7-day discussion. All these are already available on the YouTube channel. Now we begin with day 8. So starting today, that is 3rd of September till 7th of September, guys, we have another series of microbiology that is put out for all of you. And this series is again timed at the same duration, that is at 3 p.m. in the afternoon. So the post lunch slumber, and to beat that postprandial dizziness, this is a series which probably would help you. Now, if you remember, last time on day 7 of microbiology, we had started off with virology, viruses, and we finished all the different inclusion bodies, the electron microscopic images that you had to know. Now we move on to something to do with classification, what are the methods of cultivation, what are cell lines, and all the questions that you get associated with it. Do you guys remember where did we leave the last episode? In the last episode, we discussed all the mnemonics, which are the DNA viruses, amongst the DNA viruses, which are double stranded, single stranded, then amongst the RNA viruses, which are double stranded, single stranded, which have one copy or which have two copies, which are positive sense, which are negative sense, which are segmented, which are unsegmented. And if you feel this sounds like Greek to you, please get back to the previous lesson and revise all these mnemonics. So continuing with the mnemonics, here we begin with another one. Which of the following viruses has the smallest genome size? And you need to tell me the same. So the smallest genome size is in which? And out of these, the smallest one is in hepatitis B. But actually, the smallest one is not mentioned here. So if I have to actually go into the theory of it, all of you know that when I'm dealing with viruses, viruses can either have RNA or they can have DNA. It's never both. It's either RNA or a DNA. If they ask you which are the largest genome, retrovirus has the largest genome. If they ask you the smallest is with hepatitis D virus followed by hepatitis B. So now hepatitis D is the smallest, but hepatitis D was not given in the question. So my best answer in that scenario becomes hepatitis B. One more thing about the genome, now whether it is covered with an envelope or not, so whether your virus is enveloped or non-enveloped is also important a mnemonic for you to learn. So remember, amongst the non-enveloped, so usually we call them enveloped, but amongst the non-enveloped we have PAP and PARCH. What is PAP and PARCH? So DNA viruses, all the VO and O sounding ones, okay? So if I say PAP, PAP, parvovirus, adenovirus, papovavirus. So remember parvo, adeno, papovavirus. These are the PAP, these are non-enveloped. What is PARCH? Picornavirus, astrovirus, riovirus, calcivirus, hep A and E. So repeating, picorna, astro, rio, calci, hep A and E. So remember, PAP and PARCH do not have an envelope. Well guys, having read these last two mnemonics, now we move on to the next, talking about some cultivation methods. So when you're interested in the isolation of a particular organism, you are going to use suckling mice. So basically I'm asking you, animal inoculation is used for which of the following? For the isolation of what? So I hope everyone knows the answer, and the answer to this question is Coxsackie. So let me tell you a quick brief on this. All of us know that we can have Coxsackie A and Coxsackie B. And we also know that if you put Coxsackie A in a suckling mice, this is going to result in flaccid paralysis. On the other hand, if you put Coxsackie B in a suckling mice, we'll end up with spastic paralysis in the mice. And how do I learn this and not get confused in the paper? So all of you know that A comes before B. So obviously, F will come before S. So flaccid will come before spastic, A will come before B, so you'll never interchange the two. Having done this, moving on to the next one. Which of this is a primary cell line? So now I'm coming to cell lines. Primary cell line is chick, embryo, fibroblast. So for those who know your theory very well, HeLa and Vero happen to be continuous cell lines and WI38 happens to be a secondary cell line. Chick, embryo, fibroblast is a primary cell line. For those who don't know, I'm going to give you some mnemonics. Before that, I'm going to ask you another question. Human fibroblast cell line is used for what? So I hope you all remember again. Human fibroblast cell line is used for cytomegalovirus. So it's time for me to explain these cell lines to you and give you a better method of identification, right? So cell lines can be primary, secondary and continuous, and obviously continuous cell lines are going to allow unlimited divisions. That's why we call them continuous cell lines. Amongst the primary, what are the things I'll read out for you? The chick embryo fibroblast, human amniotic cell line, rhesus kidney cell line. So how have I learnt it? That when I'm talking about primary, I'm going to talk about primitive things and primates. So I'm going to talk about the ancestors. Okay, we've come from monkey. So rhesus kidney cell line. Then we are basically coming from what layers? Amnion, chorion. So human amniotic cell line. And then we also know chick and embryo, they happen to be primary, primitive. So all the primitive sounding things like the monkey kidney cell line, the human amniotic cell line, chick embryo fibroblast are primary. What all comes under secondary then? So primary we've studied. Now let's focus on this secondary one. So secondary has human fibroblast cell line. So if you ever get confused which was human amnion cell line and which was human fibroblast cell line, I think you'll get it. Human amnion cell line is going to be primary, primitive sounding. Human fibroblast cell line is secondary cell line, and this is what is used for the inoculation of cytomegalovirus. They ask you this question. Other than that, the secondary cell lines also include MRC5 and WI38. So remember MRC5 and M and W. Coming to the continuous, all the short forms. HeLa, Hep2, KB, McCoy, Viro, BHK, Chang, CILK, all the short forms come here only. So if I can repeat, all the primary primitive ones come here, like rhesus kidney cell line, human amnion or amniotic cell line, and we have the chick embryo fibroblast. In secondary, human fibroblast cell line. So this is for the growth of cytomegalovirus, and two short forms. We have MRC5 and WI38, and continuous has all the short forms. So when I came back to this question, which of the following is primary cell line, which was the most primitive sounding thing? Chick embryo fibroblast is primary. Then the short forms all become continuous, but WI38 secondary cell line. Coming to this, the human fibroblast cell line, I hope you remember human fibroblast, primary cell line or secondary cell line? Fibroblast, secondary cell line. If I would have used the word human amnion cell line, you would have called it primary. So what is human fibroblast cell line used for? For the isolation of cytomegalovirus. Now, having done this, let's move on to the next question. What is this? So interferon gamma is released by. So the answer to this question before I start discussing is CD4 T cells. One more question. Which of the following is true about interferons? Is it virus derived, host derived, inactivated by nucleases, or virus specific? So remember, it is actually nothing to do with the viruses. Guys, interferons are host derived. So that's the first thing that I want to tell you. Interferons, which we write as IFN, that is how we write interferons, IFN, these are produced by the host. They are host derived. So we always say that they are host specific. They are not virus specific. Repeating, they are host derived, they are host specific, not virus specific. How many types of them do we have? Interferons, alpha, beta, gamma, and I'm actually, before I show you the written part of it, I'm going to give you some beautiful mnemonics which will help you remember these interferons, alpha, beta and gamma. So if I ask you, from which cell does their source come? So you will say alpha, beta and gamma comes from LFT. LFT means liver function test. L stands for leukocyte. Which leukocyte? Macrophages. F stands for fibroblast. It stands for fibroblast. And T stands for T helper cell 1. What is T helper cell? CD4 T cell. And that was the question that I had given to you. That interferon gamma is going to be released by what? You'll say alpha. That's how you write it in the exam. You'll think of it. Alpha, beta, gamma, liver function test, LFT. So gamma will come from CD4 T cells or the T helper cells. Okay, having said that, now let's move on to the next one. Now we come to the next question, and that is, what is their function? So the next thing that you have to know is the function. So remember, A for A, B for B and G for G. That's how you learn it. Function wise, alpha is supposed to have antiviral properties. It is having antiviral properties. Beta is used for the treatment of a brain disorder. Which brain disorder? It's used for the treatment of multiple sclerosis. And G for G. G is needed for granuloma formation. G is needed for granuloma formation. Interferon gamma is needed for the granuloma formation in the body. And interferon gamma is used in the treatment of chronic granulomatous disease. I hope all of you remember. Chronic granulomatous disease is a deficiency of which enzyme? I know it's a pathology question, but when the exam is nearby, why segregate between chapters and topics? So you are going to tell me today in the chat box that chronic granulomatous disease is which enzyme and gene defect. I'm looking out for those answers. So how do you treat chronic granulomatous disease? Interferon gamma. So remember, A for antiviral properties, B for the treatment of a brain disorder, multiple sclerosis, G for granuloma formation, G for the treatment of granulomatous disease. The story is not over. Next, they ask you, again I'm going to make the same table, that if this is interferon alpha, beta and gamma, then what are the chromosomes that are going to code for them? What are the chromosomes that are going to code for them? And if they are interferons, they will act on some interferon receptors. Receptor for alpha, receptor for beta, obviously if they are interferons, they'll act on some receptor. So the receptors are coded by which chromosomes? So these are the two questions that they get. Number one, interferons are coded by which chromosome? Number two, their receptors are coded by which chromosome? It's a really tough one, I want you to note. So instead of learning this separately, learn it as a chart. 9, 9, 21, 21. 9, 9, 21, 21. So alpha and beta interferons are coded by chromosome 9 and their receptors are coded by 21. For gamma, just flip this 21, what do you get? 1, 2. Opposite 1, 2. Flip this 9, what do you get? You get a 6. So remember, so flip it in opposite directions. Flip it, take it up, flip it, take it down. So interferon gamma is coded by 12 and its receptor is coded by 6. I know it's a pure mugging up question, but do we have a choice? We'll have to learn it. Having said that, repeating for all of you, interferons are host derived, they are host specific, they are not virus derived, they are not virus specific. Well, having done with this one as well, now let's move on to the next set of questions, and this is to do with this. Cell monolayer is used for what? So cell monolayer is used for electron microscopy, cell lines, POC assay or plaque assay. So this is again not a question that has come in your previous years, but the topic has definitely come. And the answer to this question then happens to be plaque assay. So cell lines, monolayer cells are used in plaque assay, and I'll explain that in a while. Tell me this one now. The method of viral identification that has been shown over here, all these 6 things that you're seeing over here, same options. Are they electron microscopy? I really don't think you feel it's electron. Cell lines, POC assay, plaque assay, this is also a plaque assay. So the cell monolayer that you were talking about, this is nothing but the cell monolayer, and this is also plaque assay, which I'll tell you about. But if I show you the same picture, guys, the same violet color picture that you have over here, and I ask you, what was the stain that was used? So the same color that I've added up, I think I've given you the answer. If I ask you which stain has been used, the answer is crystal violet. So for plaque assay, which stain is used? Crystal violet. So if you knew these answers, you're really intelligent. If you did not know the answers, listen to me. Now, to know the infectivity of a virus, is the virus there? Is it not there? If it is there, then what is the quantity? So to know the infectivity, you have 2 types of methods. Physical methods, chemical methods. Physical methods can be things like RT-PCR. They can be things like electron microscopy. RT-PCR, electron microscopy. Chemical methods is what you get a question on. So see, in physical methods, you can physically see either the DNA or the RNA of the virus, or you can see the virus under electron microscope. Under chemical methods, you either have qualitative or quantitative. What is qualitative? Endpoint assay. Endpoint, it tells you virus is infective or not infective. Yes or no? Endpoint. It doesn't tell you the quantification, how many viruses, what is the number. No, nothing. Just infective, there or not there. Number 2, quantitative, which will quantify the virus for you. There are two assays that are there, plaque assay and POC assay. Out of these, you get a question on this plaque assay and look at how it's done. So you take a virus stock, any virus stock, and you are going to make serial dilutions out of it. After making serial dilutions, you add it on a cell mono layer. You add it on a cell mono layer with agarose gel. So on the cell mono layer, can you see these growths? These are known as plaques. Can you see these that have developed? Yes, these are plaques. So if you want to visualize these plaques on a cell mono layer, then what is the stain that you are going to use? You are going to use a crystal violet, and once you use a crystal violet, which is going to very nicely stain the entire cell monolayer in the background. Can you see over here? It's stained the entire cell monolayer crystal violet. And now can you easily count the can you count the plaques? Yes, so easy identification of the plaques is, and counting is possible. So definitely, since counting can be done, it happens to be a quantitative method. Well, having said that, repeat of the questions. The cell monolayer is used in plaque assay. Question number 2, this kind of plaque formation, if they show you with background stained with crystal violet, this is plaque assay. Or if they directly ask you that in plaque assay which stain is used, it is a crystal violet. Well, having done this, let's move on to the next question, and it's a simple one which brings us to the classification. Which of these is not an RNA virus? So basically, which of these is a DNA virus is what they want to ask you. Ebola, SV40, rabies or HCV. Which of these is a DNA virus? I hope you remember it is SV40. It is a DNA virus. Now guys, I'll be teaching you the entire classification, nothing to worry, but please tell me one thing. SV40 has recently been associated with a particular lung tumor. And what lung tumor is it? Lung or maybe a pleural tumor. And recently it has been associated with mesothelioma. This is a recent update and I've taught you under pathology. So SV40, mesothelioma. Remember this for the exam. And this is definitely a DNA virus. So how do I pick up DNA versus RNA? What is my mnemonic for that? So guys, there's a very simple mnemonic that goes by the name of happy. It goes by the name of happy. So what do you have under it? Number 1, please, I'll write it for you. Number 1, we have herpes viridae. So it has all the herpes sounding things. Herpes simplex virus, herpes zoster virus, human herpes virus, EBV, CMV. These are all herpes viridae. Number 2, hepadnaviridae, which means hepatitis B. Hepadnaviridae means hepatitis B. Number 3, adenovirus. A for adenovirus. Then you have PPPP. So remember, that's the mnemonic. So what is PPPP? Pox virus. Pox has variola, vaccinia, molluscum. So guys, the molluscum contagiosum, the HP bodies that you have been talking about, that is nothing but pox viridae. Then you have parvoviridae. Parvo obviously means parvovirus. So parvovirus B19 comes under it. Then one more P, you have the phage. One more P over here, you have the phage, so bacteriophage. And the last P, you have papovaviridae. Papovaviridae has a lot of combination things. See, if you break papova, can you get human papilloma virus? Papova, human papilloma virus, polyoma virus, BK virus, JC virus, and even the SV40 that we just studied. So repeating all the rare ones apart from HPV. Human papilloma virus, so papova, human papilloma virus, polyoma virus, BK, JC and SV40 come under papovaviridae. So guys, I repeat, H for herpes viridae, H for hepadnaviridae, hepatitis B, A for adenovirus, P for pox virus, P for parvovirus, P for phage, that is bacteriophage, and P for papovaviridae. Which is the largest out of these? Pox virus is the largest, which is roughly, there's a range which goes up till 300 nanometers also. Okay, so it's going to go up to 300 nanometers, and the smallest virus is the parvoviridae, which can go up as low as 20 nanometers. So parvoviridae, ultimately I realize parvoviridae is quite a loner in life. If you realize, it's got everything very minimal, very less. Do you remember, out of all the things I taught you in the last class, all the DNA viruses are double-stranded. Except, do you remember? Except parvovirus. parvovirus, and parvovirus is one strand. It is a single-stranded DNA virus. Rest all of them are double-stranded, but parvo is single-stranded. Also, hasn't parvo come across one of your smallest viruses of only 20 nanometers? So here you go. So many students have this very famous mnemonic that they like to learn, that is paro is akela. Paro is alone, so it's one of the smallest and it's one of the akela single-stranded ones that you have amongst the DNA family. Coming back, which means Ebola, rabies, hepatitis C, all of these happen to be RNA viruses, whereas SV40 happens to be a DNA virus. I hope this question is very, very clear, because everything else now you can read as per your choice, guys. Everything else, Picorna, Paramyxoviridae, Calci, Toga, Ortho, Bunya, Arena, Flavi, Corona, very important, Rio, Rhabdo, Retro, Philo, all of these now come under RNA. So my simple way of learning was DNA viruses are happy, HA tetra P, okay, HHA tetra P. Okay, having said that, now let's move on to the next question, which is something which is very basic, and I just want you to quickly revise it before I end today's class, and that is this. You have to find out what is the correct order of events for viral inflammations or infections. So attachment, generation, synthesis, uncoating, or whichever one you feel. I hope the first one you can easily rule out, because attachment has to be, this one you can rule out because attachment has to be the first one, right? After attachment, immediately generation cannot occur, some other step has to occur. So once there's a virus that comes and gets attached, after getting attached, it must first penetrate, right? So after attachment, first we have penetration, then uncoating, then synthesis, then release. So let me see this. It's better if I learn it with the help of a diagram, isn't it? So for example, let me go one by one over here. The first step of a virus is going to be attachment. The first step is going to be attachment. After attachment, it is now going to penetrate. Number 2 step is going to be penetration. Now penetration can happen by a number of things. So see, attachment is a very, very important step. Attachment is between the virus and whatever host you are considering. Next is penetration. Now penetration can happen by many methods. Penetration is either this big cell just eats up the virus. So that's a phagocytosis. One method of having the penetration is going to be phagocytosis. The second method is membrane of the virus and membrane of the host. It fuses. So that is what typically happens in HIV also, guys. Fusion of the membrane can occur. So there can be membrane fusion in HIV. Second method is phagocytosis, or you say that instead of any other virus, if bacteriophage has come, let me show you a diagram. If a bacteriophage has come, if it has attached, does the entire bacteriophage penetrate? No. It only injects its nucleic acid. So penetration just involves injection of nucleic acid, and that is what it has done. So there are many methods of penetration. That in a bacteriophage only nucleic acid will penetrate. In HIV, membrane fusion will occur. In others, phagocytosis can occur. Having said that, next is the process of uncoating. So the third one that we have is the process of uncoating. What are we trying to uncoat over here? So okay, let's see. Uncoating is that around the nucleic acid, whatever capsid is there, remove that so that nucleic acid can actually come out. For example, that whatever this capsid is there, you have to remove this capsid. But tell me, in case of bacteriophage, didn't the direct DNA come inside? Was there a capsid around it? No, the DNA has directly been injected inside. So tell me, in case of a bacteriophage, will the uncoating step be present or absent? It's absent. So remember, uncoating is removing the capsid. So I just want the nucleic acid to be free. But in bacteriophage, the nucleic acid is already free. So this is absent in bacteriophages. Now after uncoating, now you say that this is ready for the synthesis of new viruses. So I can say now we go on to something known as biosynthesis. Now guys, once biosynthesis is over, now once new virions have formed, new ones have formed, package them, assemble them. So let's take the example of bacteriophage. See, new viruses have formed. So now just package and assemble them. Make them into proper bacteriophages. Proper packaging and assembly is needed. So first, after biosynthesis, we will need assembly. After assembly, you are supposed to let them mature, and once they are mature, you can let them release. So again, repeating for all of you very clearly that after so many biosynthesis of bacteriophages have occurred, you tell them to assemble. That listen, viruses, you tell them to assemble properly, you tell them to mature over here, and then finally see they have been released. They have been released. So that's the last process. So let's repeat all of these that we've studied up till now. Let's take bacteriophage as the virus. So the number one step is attachment. Attachment over here. Number two step happens to be penetration. Number three would have been uncoating in any other virus, but not over here. It is absent in bacteriophages because it is already uncoated. There is nothing around it. Okay, after that there's going to be new synthesis. There's going to be number 4 process, and that is biosynthesis. After biosynthesis is done, means lot of products and raw material is ready, you just need to assemble them. So the next step is going to be assembly, and then you wait for them to mature. Once they are mature, they are ready to be released. And that is why I say that's the entire process of steps that are important in the viral replication. So guys, what out of this is said to be the eclipse phase? What out of this is said to be the eclipse phase? So please remember, the eclipse phase is the time between the host entry and the first virus to be released. So the time between the host entry, and the virus to be released is said to be the eclipse phase or the eclipse time. So some people call it eclipse phase, some call it eclipse time. And please remember, for a bacteriophage, it is very short, only 15 to 30 minutes. But for any other virus, it can range like a TB or any other, in case it's growing in a TB, a bacillus or any other virus, it can range up to 15 to 30 hours also. So remember, for bacteriophage virus, this is 15 to 30 minutes. For other viruses, it is 15 to 30 hours. And also depends on what is the host. If the host is a TB or the host is any other bacteria, so also depends on that. But for the bacteriophage, 15 to 30 minute question comes. Well, with that guys, we do finish our day 8 of general virology also. And if you see, we've done the methods of cultivation, suckling mice, we've done the cell lines, we've done the classification, and we've also done the replication. So as promised, our general virology is over. Now what's the plan of action from tomorrow? From tomorrow, that is from 4th of September, same time, 3 p.m, we are starting with bacteriology. And how are we going to study bacteriology? We are going to study this with the help of a mixed bag of roughly 50 questions. I am trying to touch upon most important bacteria which require a lot of images and mnemonics. So total of 50 questions, not in one day. Over the next 3 to 4 days, we'll be discussing and we'll touch upon all the important bacteria in the form of long stem and other questions that are needed. Feedback is always welcome. Homework is always being looked upon. So I hope you guys are completing the same and let me know. Thanks a lot for joining in. See you tomorrow. Have a great day.